Good evening. There we go. And I just want to make sure that those online can hear me as well. Is that a thumbs up? Okay, louder here. And at home, are they hearing us? Do I have the mics on? We have about 300 people attending this wonderful gathering today. Thank you. And again, some of those are at home with us today. So welcome. All right, I think I have the A-OK to go with sound. Okay. Well, welcome to our audience in the John J. Hemmingson Center on the Gonzaga campus, and to those of you who are on, again, our live feed to our Fall 2024 Next Generation Medicine Lecture. We're excited to have Dr. Nora Disis, from the University of Washington School of Medicine here to share her compelling research on breast cancer vaccines. And if you have seen the ads about Dr. Disis, you have seen her many accolades and different representations of institutions she's coming from, and she will be sharing that a little bit with us today. My name is Dr. Julie Walter. I'm the Dean of the School of Health Sciences here at Gonzaga University. And for those of you who don't know about our UW School of Medicine GU Health Partnership, I just wanted to share just a tiny bit about that because this is one of the sponsors for tonight's event. So in 2016, the University of Washington School of Medicine and Gonzaga University united to advance the health of communities throughout Eastern and Central Washington through medical education and research. Each institution has a legacy of graduating innovative, community-focused students, and through our partnership, we leverage this strength. Since we signed on the dotted line 8 years ago, the partnership has grown. It now includes direct ties between UW Medical School and the GU School of Health Sciences, which houses about 1,000 undergraduate and graduate students in areas such as nursing, human physiology, public health, as well as our partner undergraduate programming in our other GU college, such as our new neuroscience program, as well as partner faculty in areas like sociology. So we're really excited because there are co-appointments of faculty across these units where our students get to work with faculty who also serve our UW med students. So it's a really unique and exciting partnership. Our partnership home includes a state-of-the-art, education building where UW medical students, GU human physiology students, public health students, and soon even UW dental students in the RIDE program, which stands for the Regional Initiatives in Dental Education, and perhaps even new health science programs in the future where we come together and share resources in the Health Partnership Building, which is that big white building that you might have driven by today. So additionally, in the Spokane Inland Empire Railroad Building, which we call the SEER building, across the parking lot, there is a program which is the UW's MedX Northwest program, which houses our physician assistant students, and the UW's Spokane Center. So we're really excited about the growth of our partnership, and we look forward to thinking about new and innovative ways to be going about health education, research, and community partnerships in the future. So with that, I want to hand the mic over to my wonderful colleague here, Dr. Podick, who will say a little bit more about our wonderful speaker tonight and welcome you to this program. Thank you.
Hi there. Thank you all for coming out tonight. It's a beautiful fall night, and we're just glad that you decided to come spend part of it with us. So I'm Daryl Podick. I'm an internist and geriatrician here in town. I work with the internal medicine residency program. I'm also the Associate Dean for the School of Medicine here in Eastern Washington and the Chief of Medical Education for our partnership. Before we get started, I wanted to thank our sponsors for this evening, and those include the Spokane County Regional Health District, the Spokane County Medical Society, and the University of Washington's Population Health Initiative. Just want to give you a brief overview of what we're going to do tonight. Dr. Disis will present her work and her very exciting work over the next 40 to 45 minutes. We'll have a Q&A session after that, and we'll allow some of our online participants to submit questions as well. And then we'll wrap up by about 8 o'clock. And so let me tell you a little bit about Dr. Disis. It's just such a pleasure to have her with us tonight. When we first started this Next Generation of Medicine series, lecture series, back in 2017, Dr. Disis was our very first speaker. At that time, we were thrilled. She talked to us about what was happening in the world of cancer vaccines, and she's back tonight to really to share updates and let us know what's new and what we can be hopeful about in terms of cancer vaccines. Dr. Disis is incredibly busy, and we're grateful that she made time in her schedule to be with us tonight. She told me she drove 100 miles an hour on the interstate today to get here, so she's kind of my hero. Dr. Disis did her medical school at University of Nebraska. She went on to do an internal medicine residency at the University of Illinois in Chicago. She did her oncology fellowship at University of Washington. She's currently a professor of medicine in the Division of Hematology and Oncology at the University of Washington. She's the director of the Cancer Vaccine Institute. As you can see on the slide, she's the Helen P. Slonaker Endowed Professor of Cancer Research. She's also editor-in-chief of JAMA Oncology and deputy editor of the Journal of the American Medical Association. Her research focuses on immune-based therapies for common cancers such as ovarian cancer and breast cancer. She's dedicated to translating her lab discoveries into really benefiting the lives of patients across our nation and across our world. She holds several patents for targeted cancer therapies and diagnostics. And just last night, she got an award as the UW Inventor of the Year. So she received that award last night, and just a little preview of what we might expect. The person that won that award a few years back today got awarded the Nobel Prize from UW. So we're hoping that Nora will follow in those footsteps. Please join me in giving a nice warm welcome to Dr. Disis.
It's great to be back here in Spokane and at Gonzaga, and I look forward to raiding the bookstore tomorrow before I go home. So today I'm going to talk about our work on cancer vaccines and especially how close we are to an approval for a breast cancer vaccine. And of course, I always like to start in talking about the importance in general of vaccines, because vaccines have really had a profound impact on human health. And I think our thinking about vaccines are all caught up in the COVID vaccine, right? And all the murky thoughts that we have about the COVID vaccine, that sometimes we forget that vaccines have really changed the face of human disease. And when you think about it, a lot of the diseases that used to kill us 50 years ago, 100 years ago, are no longer on the face of this earth for the most part because of vaccines. So diseases like polio, smallpox, the measles are literally not here anymore because of vaccines. And these diseases would be totally gone if everyone would go and get their vaccines. So many diseases that used to cause us serious harm or death 50 or 100 years ago are not here anymore because of vaccines. So what are vaccines? What do vaccines really do? How do they work? We talk about them as if they're one drug, but really they're a complex component of many different things wrapped up in one package. Vaccines train the immune system to recognize and destroy foreign threats. So basically, you have immunogen, or an immunogenic protein that's teamed up with an immune stimulant that we call an adjuvant. And this is put into a vehicle that can deliver this immunogenic protein and an adjuvant to the skin or into the mouth or into some organ that allows the immunogen and the adjuvant to be presented to the immune system. So in our case, we use DNA or mRNA as that vehicle to deliver the immunogenic protein and the adjuvant. And the immune system then receives that immunogenic protein and adjuvant, and the immune system begins to recognize it, and it gets primed so that the immune system can then go throughout the body, and whenever it sees that protein, whether that protein is a foreign germ or that protein is part of cancer, the immune system can then respond to it. And those trained immune system cells will then search for wherever that protein is in the body, and it will destroy it. So in the case of a virus, when you get immunized against a virus, and then the virus comes into the body, the immune system is already pre-trained to see that virus and destroy it. So when that virus enters your body, you've already killed that virus before you even know you've gotten the virus. And that is the hope eventually for a cancer vaccine, that if you have trained the immune system to recognize a cancer-causing protein, that when that cancer-causing protein begins to arise, your immune system has already been trained to kill that protein, get rid of that cancer, before you even know that that cancer has started to develop. That's the holy grail of cancer vaccines. That's what we're working toward, to be able to develop cancer vaccines to prevent you from developing cancer. So how are we now at the point where we think we can develop vaccines to prevent you from developing cancer, or getting a vaccine that can prevent you from having your cancer come back if you've had cancer? Well, we're at a tipping point for cancer vaccines. You may think we are also, because you can't help but open a newspaper, the Wall Street Journal, the New York Times, without reading some article that says, we've got a vaccine for melanoma, we've got a vaccine for non-small cell lung cancer, a pancreatic vaccine, we're curing pancreatic cancer. How many people in the last couple of months have read some article that there is a vaccine that is curing someone with cancer? You know, I've read some. Well, where there's smoke, there's fire. Lots of people, lots of researchers have been developing these vaccines that are starting to show results. And we are at a tipping point for three major reasons. We know what type of immune response we need to eradicate cancer. And it's totally different from the type of immune response we need to get rid of germs. To get rid of a germ like COVID, we mostly need neutralizing antibodies. That's what you get when you get a COVID shot. For cancer, we need type 1 T cells, lymphocytes. These are living cells that can actually secrete cytokines that cause cytotoxic T cells, another type of living cell, to come in and eat the cancer like a Pac-Man. For those of you who lived through the Pac-Man era, that's not going to be you. Look it up on Google. So these are living cells that actually have the capabilities of eating and killing a cancer cell. So you need type 1 T lymphocytes. And all the adjuvants and different types of mechanisms for creating vaccines for infectious disease are not the types of mechanisms and adjuvants we need to create cancer vaccines. And that's taken us a while to figure out. Secondly, the next tipping point is we now know hundreds of immunogenic proteins that are expressed on cancer cells, and we didn't know that before. So we can literally tailor-make cancer vaccines for specific types of cancers. Hormone receptor positive breast cancer, triple negative breast cancer, HER2 positive breast cancer, specific types of colon cancers. The revolution of genetic sequencing of the human genome and now the tumor genomes have allowed us to really identify with precision the proteins that become overexpressed and immunogenic and the driver mutations that are present in specific cancers. And that has really revolutionized the design of cancer vaccines for literally any different type of tumor. And the final tipping point for cancer vaccines is we now have safe and effective vaccine technologies. And I will say that those vaccine technologies are nucleic acid-based technologies, DNA and mRNA vaccines. And the reason why these vaccines are so effective is that they allow your skin cells to be transfected to take over the immune machinery within the individual and allow the patient's own genetics to dictate how those immunogenic proteins are processed and presented, and everyone's own individual immune system machinery can present the antigen the way that individual's immune system wants to see it. And it takes away the need for people to be re-engineering the antigen outside the body to try to figure out how to make that antigen fit into that patient's what we call MHC, or a genetically dictated immune receptor. The way that we deliver the vaccine allows the immune receptors to have the antigen encoded inside the body and presented by the MHC inside the body. So if I take a peptide and I make it in a machine and I inject the peptide into the skin, you may make an immune response that's maybe this high because the peptide has to bind exogenously to your MHC receptor that's present on the surface of your skin. But if I give you a DNA vaccine, the DNA will get inside the skin for a short period of time, and that DNA will be processed inside the skin, and your MHC molecules will allow the DNA to chop up and process the antigen the way your own immune system wants to have that antigen processed. And the immune response will be logs-fold higher. And we've learned that in the last 10 years, and COVID has taught us that. So these three things, knowing the type of immune response needed to kill cancer, the hundreds of tumor antigens, and these tremendously powerful vaccine technologies have resulted in a revolution of new and effective cancer vaccines. And we're taking these vaccines to clinic. Now, there are two types of antigens in general when we talk about cancer vaccines. One type of antigen are mutated or foreign antigens. And when we talk about tumor mutations or foreign types of antigens, there are really two types. Some cancers are caused by viruses, such as human papillomavirus or hepatitis B virus. And of course, viruses appear dangerous to the immune system. So those antigens are already seen as foreign to the immune system. And tumors that are caused by viruses, like hepatocellular carcinoma or cervical cancer, these are inherently more immunogenic. Or tumors that are highly mutated, such as melanoma or kidney cancer. These tumors can be treated with vaccines that are created by putting together multiple mutations. And basically, you can take a patient's tumor, figure out all the mutations in the tumor, create all these mutations and encode them in mRNA, and tailor-make a mutated vaccine specifically for that patient. These mutated vaccines are very effective in some patients, but they're incredibly expensive and they must be made for these individual patients. But they're highly effective because these mutations are seen by the immune system as foreign antigens. They give the immune system a danger signal. But for the vast majority of patients, most antigens are not mutated. They're non-mutated. And these antigens are self-antigens that become highly upregulated or overexpressed. So when you think about a cancer cell, it's really a self cell that starts growing out of control. So many of the proteins in that cell, like growth factor receptors or cell cycle proteins, instead of one of them in the cell, suddenly you have 10 of them in the cell. So they're markedly, aberrantly upregulated. But they're not mutated. They're the same self-protein, just found in abundance. This doesn't give a danger signal to the immune system. The immune system sees it as abnormal, but sees it as self. So the immune system sees it as potentially dangerous, but the immune response that the immune system puts forward is an antibody response that does not give the killer response. It gives an immune dampening response because the immune system does not want to cause autoimmunity. So if I took a patient with melanoma and I tested their blood cells, I could find killer T cells just endogenously. Their tumor is already educating the immune system to make those killer T cells without a vaccine. But if I took a person with breast cancer and looked at their blood, I would find antibodies. I would find no evidence of those killer T cells because the immune response they're creating against their tumor is one that's telling the immune system, don't respond to these antigens. So one of the things that I really worked on, since we're looking at all the tumors that really are those where the antigens are those non-mutated antigens, it's how can we make these antigens appear more dangerous to the immune system? And one of the things that we found was within the natural sequence of these non-mutated tumor antigens were sequences that selectively elicited a type 1 T cell response. And these were found in very small numbers. And sequences that selectively elicited a type 2 T cell response, that type of T cell response that said, turn off that type 1 T cell response. And these were found in very high numbers. And what we did was we created methods of artificial intelligence where we could take that non-mutated sequence and use artificial intelligence to help us pick out those rare type 1 selective sequences and pull them out and identify them and separate them from those type 2 sequences so we could create type 1 selective cancer vaccines. So we create methodologies where we could identify non-mutated proteins that were cancer drivers for a particular type of cancer, and then predict which protein segments had that type 1 T cell response. We used artificial intelligence to create a barcode that allowed us to figure out that effective cancer vaccine. And then now literally with DNA technology, we could simply create DNA segments, encode them into DNA, and create DNA vaccines and put them into vaccine vials and get them ready for injection into patients. And this revolutionized what we had been doing before. Something that took us 15 years previously, we had streamlined into being able to do within 18 months. And so nucleic acid-based vaccines were the key to being able to go from a thought into a tangible vaccine within 18 months. And these were adaptable to multiple antigens because cancer isn't caused by one protein, right? So it's very rare that you find a tumor where you can immunize against one protein, like immunize against a COVID protein and be able to stop COVID. Even when you look at a flu vaccine, you're immunizing against multiple different types of flu. So you have to immunize against multiple different types of proteins to be able to stop a cancer. And DNA and RNA, like I told you, are superior in the generation of those Pac-Man-like toxic T cells that are able to eat the cancer. And DNA is stable over time at higher temperatures. So this truly is a vaccine that you can put in your pocket and take it into a foreign country like Africa and be able to take it across the world and treat patients where they are, wherever they are. And one of the beautiful things about DNA and sometimes RNA is that it can persist at the injection site over a period of time, so it can auto boost. And that creates memory T cells. So if you're contemplating trying to create memory that will allow people to potentially protect themselves over an extended period of time from the development of cancer, that development of immunologic memory really is critical. They're very low cost. Unlike creating personalized vaccines for every individual patient, off-the-shelf DNA vaccines are $30 per shot. And like I said, they're very easy to manufacture. So now, with this in mind, we're developing vaccines for the most common and deadly cancers on colon, bladder, prostate, ovary, lung, and breast. So you might ask, why these cancers? Because these are the cancers that are responsible for 50% of all cancer diagnoses and 50% of all cancer deaths in the United States and pretty much the world. And what are we developing DNA vaccines to do? We're developing them to treat cancer, and we're teaming them up with immune drugs. And I'm not going to talk about that today because that's not our main focus. But we're developing them to prevent cancer recurrence. That is one of the biggest causes of death from cancer. We're very good at treating cancer. In a lot of cases, we're not that good at making cancer not come back. But we're also spending a lot of effort figuring out how to make cancer vaccines that will prevent cancer from even occurring in the first place. So I want to show you just 3 examples of this concept of preventing cancer recurrence and preventing cancer from occurring in the first place using breast cancer as an example. And this is our roadmap for breast cancer vaccines. The goal of preventing death from breast cancer. And I think we're going to get there within the next 10 years. Not just our group, but several groups across the United States are working on this as well.
Can I put new batteries in your microphone really quickly?
Sure.
Your laugh will continue to get.
So the first story is going to be about preventing breast cancer from recurring. And I'm going to show you kind of our hallmark antigen, which is a 1 antigen vaccine with a very rare instance of a driver protein that really lets you use 1 antigen. Then I'm going to show you an example of a multi-antigen vaccine that may allow you to prevent breast cancer from even starting in the first place. And then I'm going to show you an example of a brand new concept of reducing breast cancer risk. So let's talk about a vaccine to prevent breast cancer recurrence. And this is a vaccine targeting a protein called HER2. HER2 is a non-mutated protein. It's highly upregulated on about 30% of all breast cancers. In fact, if you have a HER2/neu positive breast cancer, it's defined by the fact that it upregulates HER2/neu. When your breast cancer upregulates HER2/neu, you go from having a single copy of HER2/neu in that breast cancer cell to having multiple copies of HER2/neu. And when you look at it underneath a microscope, you may have 100-fold more HER2/neu expressed on that breast cancer cell compared to a normal cell that has HER2/neu on it, like a skin cell or a gut cell. So we found, using our methodologies that I showed you, that most of the Th1 selective epitopes, the ones that could stimulate that type 1 T cell response, lay inside the HER2/neu protein in the intercellular domain. So we created a HER2/neu intercellular domain vaccine. And we ran a phase 1 clinical trial in stage 3 and stage 4 HER2/neu positive breast cancer patients. We enrolled 66 patients that had no evidence of disease or had bone-only metastases. And they received one of 3 doses, 10 microgram, 100 microgram, or 500 microgram. And we gave the vaccine intradermally once a month for 3 months, like you got a COVID shot. We gave an adjuvant called GM-CSF, which stimulated dermal dendritic cells, which are very potent antigen-presenting cells that live in the skin. So they really stimulate the immune system. And we looked at safety and the immune response. And since this was the first DNA vaccine targeting one of these non-mutated antigens, we were able to look for clinical response because we followed the patients for quite a long time, up to 10 years. We found that all 3 doses stimulated the immune system, but the intermediate dose, the 100 microgram dose, was the most potent in stimulating high levels of HER2/neu specific immunity. That's the red bar. The interesting thing was, at 10 years of follow-up, over 50% of the patients hadn't relapsed their disease. And about 85% of the 100 microgram dose patients are still alive 10 years after immunization. In fact, all of the stage 3 patients are alive today. None of them have recurred their disease. This vaccine has been licensed by a commercial partner and now is in randomized clinical trials. So we hope this will be the first breast cancer vaccine that will be approved for use. And there are other HER2 vaccines by other companies that are also rushing to the market. This vaccine is now in what's called a basket trial. It's being tested in different types of HER2 positive cancers: gastric cancer, lung cancer, uterine cancer. So we hope that we can find other cancers where this will be effective. And it was safe. There was no autoimmune disease generated with this vaccine, and it was highly effective in terms of generating long-lasting immune responses. So this is an example of preventing disease recurrence. And I think this is where we'll begin to see many vaccines coming into the market in what's called the adjuvant setting, preventing cancer from returning. As we go forward now with many of our vaccines, this is kind of our sweet spot for our other diseases, creating vaccines to prevent disease recurrence. We started contemplating if we were going to create a vaccine to prevent cancer, how would we do that? If we were going to prevent breast cancer, what would be our approach? So we started thinking about targeting cancer stem cells to prevent breast cancer. So what is a cancer stem cell? A cancer stem cell is the cell in a cancer that really holds the malignant potential of that cancer. So if you have a cancer, this is the cell that very early in the cancer loses its anchorage independence. So one of the things that has come about in cancer is that we have circulating cancer cells. And even at very early stages of cancer, in many patients, we can find cancer cells floating around in the blood. And these cancer cells, under the microscope, look very angry. They've already started a pathway called epithelial to mesenchymal transformation. And these cells are resistant to therapy. They are not responsive to chemotherapy. They're not responsive to radiation therapy. And these cells are the cells that cause early metastases. And in some cancers, let's say in breast cancer and triple-negative cancer, these cells can be enriched. And unless you remove them with surgery, these are the cells that are going to cause disease recurrence. So we went into the literature and we looked at, did a deep dive on the genetics of cancer stem cells. We identified several proteins that became highly upregulated early in cancer stem cells. We identified proteins that were associated with the cancer stem cell, became highly upregulated, were known to be immunogenic in cancer patients, were associated with a poor prognosis. And we found several that were on different pathways in the cancer stem cell. And we used our rubric that I showed you to identify Th1 selective epitopes from those cancer stem cells. And we came up with a vaccine composed of 5 proteins involved in the cancer stem cell pathway, and we created a vaccine called StemVac. We tested that vaccine extensively in animal models and showed that we could prevent the development of triple-negative breast cancer. And in this mammary tumor model, transgenic model, when we immunized the animals when they were 8 weeks old, we showed that we could even prevent the development of breast cancer in animals that were genetically engineered to develop breast cancer. And the animals lived till old age. They died of old age before they developed breast cancer. The animals didn't have any untoward effects from vaccination, and with this data, we were able to go to the FDA and get approval to run a phase 1 clinical trial of StemVac. So we went into an advanced stage breast cancer patient population again, stage 3 and stage 4, this time hormone receptor positive breast cancer and triple negative breast cancer. And we went into an advanced stage population because this was a non-mutated tumor antigen vaccine, and the proteins we were immunizing against were things like CD105 and other biologically very important proteins, and there was concern about safety. We enrolled 30 patients, 10 patients per arm, and they received 3 doses, 150, 300, 600 microgram intradermally once a month for 3 months. And this time we did boosters at 3 and 9 months, and we looked for safety and an immune response. It was a much shorter study. And here you can see the intermediate dose, which was again a winner. And the boosters mattered. The patients developed immune responses to all 5 antigens in the StemVac vaccine, and the level of immunity they achieved was as high as 1 in 300 of their circulating white blood cells was a T cell, a type 1 T cell specific for a StemVac protein. Very high levels of immune response to StemVac. In fact, when you looked at the responders, there was only 1 non-responder. From baseline to maximal response, they boosted their immune response 500-fold. During the time of this study, which was 3 years, we had no one relapse their disease. So StemVac now has gone on to 2 phase 2 studies, one in triple negative breast cancer, which is fully enrolled. We're in the observation period now to see if we can prevent disease recurrence. And we have one ongoing in non-small cell lung cancer. And under development is looking at StemVac in terms of preventing cancer in people who are at genetic high risk. In fact, this graph shows you all the diseases that are associated with tumors that are highly enriched in cancer stem cells. So it almost looks like a familial genetic high-risk pattern, Lynch syndrome or genetic high-risk syndromes. So this could really offer people who are in genetic high-risk families something other than getting constant screening, or in addition to constant screening, a potential of protection. So we hope to be designing some trials to see whether StemVac can offer something more than just, every 6-month MRIs to this type of patient population. So my final little story is moving cancer vaccines even further and kind of flipping over to all those epitopes that we've been collecting that give the immune system the turn off signal. So along with identification of all those epitopes that give that type 1 selective response, we've been collecting epitopes that give the immune system that type 2 response that turn off the immune system. And one of the things we know is that one of the impetus for the development of cancer over the long term is chronic inflammation. And one of the things of chronic inflammation, or one of the causes of cancer that's associated with chronic inflammation, is obesity. And obesity has become an epidemic. By 2040, I think something like 50% of all America is going to be morbidly obese. And one of the problems with obesity in some people is they develop metabolic obesity, and that's associated with diabetes and hypertension. And in metabolic obesity, you have metabolic dysfunction. And this isn't because you have too many adipocytes. What happens in metabolic obesity is that your adipocytes grow bigger. And when they grow bigger, they become stressed, and the blood vessels become stressed, and hypoxia develops. When hypoxia develops, adipokines begin to be secreted, and these are cytokines that cause immune cells that are present around those adipocytes to give danger signals to the immune system. Now, when you get danger signals, what happens? You get a type 1 immune response. So those Pac-Man-like cells come into those adipocytes, and they should not be there. And what happens is those immune cells begin to present antigen to those T cells. And the antigens are upregulated self-proteins. Those upregulated fat proteins are immunogenic, just like those upregulated cancer proteins. So you have those T cells being immunized in a type 1 way by those immune cells that are present in that stressed out fat. And what happens is those fat cells begin to get dysfunctional, and the T cells get dysfunctional. When the fat becomes dysfunctional, you get diabetes. When the T cells get dysfunctional, you lose the ability to surveil for cancer. And over time, that chronic inflammation creates genetic screw-ups that never get repaired, and you get cancer in the fat areas of breast, colon, endometrial, prostate. These are the fatty areas where you get cancer. And that's why we have these cancer-associated proteins. So we ask the question, can we take type 2 T cells that are so effective at turning off type 1 T cells and create a vaccine directed against these upregulated fat proteins and drive these type 2 T cells to the fat and turn off this type 1 inflammation? So we flipped the switch. We identified these type 2 epitopes. We created a type 2 T cell that we called adipocyte-directed vaccine, ADVAC, type 2 adipocyte-directed vaccine. And we decided to immunize fat mice. So what we did was we took mice, we fed them a McDonald's diet, and the first thing we did was we tackled diabetes. So at the time that the animals became fat, we gave them ADVAC. And in one panel, you can see a glucose tolerance test where we have the control animals in blue and the ADVAC animals in gray. And when we gave them the glucose tolerance test, you can see that the ADVAC immunized animals, even though they didn't lose any weight, improved their glucose control almost to the level of a lean mouse. So the next thing we did is we took that transgenic mouse that we were able to give StemVac to and prevent the development of breast cancer. When we made that transgenic mouse fat, their breast cancers grew a lot quicker. But we did the same thing. When they became fat, we started immunizing them with ADVAC, or we gave them a control vaccine. And you can see the control animals, all of them develop breast cancer. But just giving them the ADVAC vaccine to drive the type 2 T cells to their breasts was able to prevent the development of breast cancer in about 60% of animals. And the animals that did develop breast cancer, their tumors grew much slower than the animals that did develop breast cancer. So this is the first vaccine to lower the risk of breast cancer and obesity. It's a vaccine directed against a risk factor. So we're in the process right now of making sure that the vaccine doesn't have any untoward effects. And our first clinical trial will be looking at the vaccine in terms of people who have rising blood sugars, just to see if it can be a vaccine to reverse diabetes. We think it's a metabolic corrector vaccine that eventually would be able to be a treatment for non-alcoholic fatty liver disease or increasing hemoglobin A1C, but eventually would take away the risk of those obesity-induced cancers. So will we have a world without breast cancer due to vaccines? I think we're well on our way to do so. We know how to categorize people based on their breast cancer risk. We will have vaccines that we will use for therapeutics, vaccines that will team up with other immunomodulators that we will use for treatment. Those are in the works right now. We will have vaccines that we will use in the adjuvant setting to prevent disease recurrence. I think those will be approved for treatment within the next 5 to 8 years. We're working on vaccines that we will use for cancer interception. Those are vaccines I haven't talked about to treat ductal carcinoma in situ or atypical ductal hyperplasia. We will have vaccines that will be used in genetic high-risk patients. There are clinical trials out there right now, not only with our group, but with other groups that are immunizing patients who are BRCA1 and 2 gene carriers. There are clinical trials on clinicaltrials.gov. And finally, I've shown you a new area, vaccines that lower disease risk. This is the first, but I'm sure that there will be more to come. I'd like to end with a slide of the UW Cancer Vaccine Institute. This type of research does take very large research groups, and our translational research team here did all the work that I showed you here today, and I'd love to answer any questions you might have about tumor immunology or cancer vaccines in general. So thank you very much. Yes.
The profile of the vaccines, is it pretty minimal?
The side effect profile is very similar to any other vaccine. Probably 60% of patients get injection site reactions, fatigue, myalgia, nausea, and headaches. Those are the big ones, and they occur in the first 72 hours after immunization. We have seen the development of lymphopenia, which is a drop in lymphocyte count, mostly in the studies we do where we're enrolling the more advanced stage patients, and that is transient and goes away on its own.
A question about the HER2 target. Healthy cells express that too, correct?
Yes.
So are there any concerns with potentially long-term use of vaccines of, affecting some of those healthy cells? And maybe are there targets in the body, kind of like with chemo? I know that's something different, but you get worried about cells that rapidly divide with side effects with that?
We've followed people pretty extensively with HER2. We've done peptide-based vaccines and protein-based vaccines and DNA-based vaccines now with HER2. And we followed people out 20 years. We have not seen any evidence of autoimmunity. Having said that, they have with Herceptin. Herceptin has caused cardiac toxicity and that has been due to off-target effects with HER2/neu on the heart. And there has been toxicity with CAR T cells to other tissues that have expressed HER2. Now, the difference between what we're doing and what they're doing is that those other modalities have a higher affinity. They're more engineered to have a higher affinity for the HER2/neu receptor than the T cells that we're generating. We know that the endogenous T cells that we're generating by immunization have a lower affinity for the HER2/neu receptor than these. That's why, you can put a CAR T cell in and they can shrink a tumor, whereas we can't take our T cells out, grow them to great numbers, put them back in, and have them like totally shrink a tumor down like a CAR T cell, because ours aren't as avid for the receptor as those engineered T cells are. The end game is our T cells probably work slower over a longer period of time, have less off-target effects. Theirs work faster, but have more off-target effects to tissues that have lower levels of expression. Yes?
It looked like you guys, sorry. It looked like in the trials that you guys have done so far that you were looking at mostly stage 3 and 4 for your trials. Were you targeting any specific stage or differentiation, or was it kind of across the spectrum? And were you using it in conjunction with other neoadjuvant therapy or adjuvant therapy, if that's necessary?
Yeah. For initial trials, we target stage 3 and stage 4 because of the FDA. So they generally did not let us go into earlier stage disease for those trials. Subsequently, they've been letting us go into earlier stage disease now that they've got a little bit more background on these mutated antigen vaccines. So we've got a vaccine that targets HER2/neu, IGF-BP2, and IGF-1R that we put into stage 1, 2, 3. So in our current newer vaccines, they're being more lenient with letting us go into more appropriate patient populations for what we're aiming for. That vaccine is a cancer interception vaccine that we're looking to treat abnormal mammograms with. That vaccine is also one that we have in the neoadjuvant setting. So we've got a clinical trial where we're giving it with patients who are HER2 positive, who are getting neoadjuvant chemotherapy, where the end game of the clinical trial is a pathologic complete response. So we're giving it along with chemotherapy because we're trying to integrate the vaccine into standard of care treatment. So we're trying to start out the vaccine where we think the final home for the vaccine would be.
Thank you.
Yes?
You mentioned that the nucleic acid-based vaccines can auto boost. However, in some of your clinical trials, the patients were boosted anyways. Does this auto boosting kind of increase the time between boosters, or is the boosting done in the clinical trials just for efficacy purposes?
The boosting issue is a big issue. So, for example, if you read or went to some of the papers that we published, DNA persists in the skin. I think it's a benefit. mRNA does not persist like DNA does. That's why the COVID vaccines, you have to get them so frequently. They're not good at creating immunologic memory. That's why we are constantly getting COVID vaccines. In fact, they're coming out with a DNA COVID vaccine. But the dose of the vaccine in DNA matters. So if you went to our publications, the 600 microgram dose, it persists too long. And so it continues to expose the activated T cells to antigen, and it inactivates the T cells. So it gives them too much antigen. So if you looked at the paper, they get a good, after 3, they get a good boost, but over time, the immune response goes down. Whereas the intermediate dose, they get a boost, and the immune response persists. And it was the same thing for the StemVac vaccine. The intermediate dose, the boosts helped, but for the highest dose for StemVac, the boosts made it worse. So we've done 3 dose studies now with different vaccines, and it's that 100 to 300 microgram dose, which is the sweet spot for DNA. So how much we'll have to boost, our studies now are not so much looking at the short-term boosting. I think we've got the 3 and then like 4 months a year. It's really going to be looking at long-term. So we're now, as we follow people for the 5 years, 10 years, we're asking people if they'll be willing to give us blood every 6 months. And some of the people who are doing those personalized vaccines have shown that this persistence of the immune response is really correlating to the clinical outcome. And they're using mathematical modeling to predict persistence, but we would like to actually see if we could measure that. Yes.
I'd like to follow up on a question with that predictive response. So my understanding is that estrogen-fed breast cancer generally comes back in the 10 to 20 year range if it's going to come back, and it sounds like you tested it for 10 years and you had a good response. What is your thinking about that 10 to 20 year period? And this is a very personal question because I'm a stage 4 triple positive cancer survivor. So tell me there's hope.
Yeah. Well, you know, with HER2/neu, we don't really know. I mean, HER2/neu is a great unknown right now. A lot of that 10 to 20 year is really based on hormone receptor positive disease because it has this concept of dormancy. And this is kind of a new concept, so they're rewriting all the, you know, these long-term outcomes. We're following patients. I mean, we have patients who've been in our vaccine studies 25 years. So every year we ask people to come back to Seattle. So we have these patient reunions, and we try to get them to come back for a party, and then we ask them if we can have their blood. So we're trying to collect that data on people and see. We've always wanted to have a booster study, and we're trying to figure out how we can do that. Our IRB, you know, is not so open to that, you know, this long-term open booster study. So we're trying to figure out how we can work with them to see if we can do that. We've had a smaller booster study, but it's a critical thing to try to build that in. I think we're going to have to just make a study where as long as you can come, just build in these boosters, just just have these studies open forever and say, every 6 months you can have a booster. We're working with them to see what they'll accept. But it's our critical question right now is boosting. And it's our patients' critical question too.
As these therapies are introduced, how do you expect that they'll interact with the current paradigm, surgery, chemotherapy regimens?
In oncology, there is no current paradigm. I can tell you that. Honestly. I mean, right now they're talking about, there's a clinical trial right now for DCIS where there is no surgery. It's watch and wait. It's prostate cancer. Watch and wait. I mean, every disease we're rewriting what we're doing.
Do you think that you're going to be able to apply any of this kind of vaccine stuff to the developmental cancers like medulloblastomas and things like that?
I hope so. I mean, I have a cancer I'm very interested in, DIPG, and I have in my mind this DIPG vaccine that I desperately want to get off the ground. And I think this approach is a good approach for that, because these are bland tumors that are not immune infiltrated. And, we do know that these CNS tumors, the CNS is wide open. You can have trafficking T cells there. They've got other problems in terms of immunosuppression, but I do think that there's hope for being able to get T cells up to the brain, and I do think it's an unmet clinical need, and especially for kids. People aren't thinking about kids enough, in my opinion.
On your vaccines that prevent recurrence, what is it about the vaccine that activates the recognition that the body and immune system isn't able to recognize on its own to prevent reoccurrence in itself?
Well, I can tell you that one of our other big interests, and it really emanated from our research in looking at these epitopes, is that in cancer, your immune system, everyone used to think that cancer patients, their immune system doesn't work very well. That's what the problem is. And that's not the problem. Their immune system works great. The problem is they've stimulated the wrong type of immune response for a variety of reasons. So you are not walking into an empty slate. You're walking into a cancer that started growing, and the immune response immediately started generating the wrong type of immune response that's facilitating cancer to grow. So you've got a cancer, it's growing, you have an immune response, it's doing its thing, helping the cancer to grow, and you're behind the 8 ball. So what are you going to do? So now you have an area away from this area, and you have an opportunity. If you have a big, bulky tumor, there's no way you're ever going to get on top of that tumor. That's where everyone went wrong in the first place. When people first started doing cancer vaccines and they thought, I'm going to take a cancer vaccine and I'm going to generate an immune response here and try to boost it, and I'm going to take a big bulky tumor and that immune response is going to get on top of this. No way. You have to debulk this thing to the point where you can't see anything, and maybe you have a chance. Like when you think about it, with a foreign antigen, like a rabies vaccine, in a therapeutic setting, you maybe have a chance if you get that person in after they've been bitten in the first 24 hours. That's what we're talking about with cancer. So if I have a patient who they're totally free of disease, but they're at very high risk of relapse, that's when I can take a vaccine that I know is causing the right type of immune response. I can immunize. I can try to get up to that 1 in 300 white blood cell is a T cell recognizing their tumor. That's 1 in 300 is like an active viral infection, you know? Then I have a chance that I can overwhelm the body with that immune response that I know might be effective. Maybe then I can get that sterilizing immune response, that all those type 2 T cells that are there, I can overwhelm them. Because if there are cells there, those cancer cells, and I'm like 10 to 1, those type 1 T cells, whatever innate immune cells that are there are going to get overwhelmed, and they're going to start getting those cytokine signals that are those type 1 signals, and they're going to go, oh, no, we're getting the foreign signals. Oh, too many foreign signals. Okay, this is danger. This is danger. You can change the tumor microenvironment. It's all checks and balances, right? So you can change it. You just have to have more of one than the other. So it is possible. But you can't walk into the war when all the warriors are playing for the other team. And that's kind of the fourth tipping point, is understanding that we can't use cancer vaccines to treat these. You know, one of the guys who was the leader in the field still believes that if you can't take a cancer vaccine and shrink a tumor, that cancer vaccine shouldn't move forward. I think most of the field, you know, is like, no. But, you know, we're really talking about, if you believe in therapeutic cancer vaccines, which I think that's still debatable, you're really talking about micrometastatic disease in terms of recurrence. But it's really a war of the tumor microenvironment.
I think we have a question in the back, but before we do that, I just want to make an announcement for those of you online. If you want to submit a question, you can please type it in. We're monitoring that, and we'll get your questions answered for those of you online as well. So go back to the question in the back.
I have a question about just the breast cancer clinical trials. Do you have any idea who some of the ages of some of the youngest patients that received the vaccines?
I would say that most people cut it off at 18. I would say routinely people say 18. And I think that's like a boilerplate. Is that your question?
Going back to prevention, before the cancer happens, I'm just thinking of some of us that might have daughters or grandchildren in the future that might have very high risks. And you think, okay, should maybe we start getting vaccines at a very early age?
Yeah, I think these preventative breast cancer vaccines, I don't think they'll ever be given as childhood immunizations. I think that there will be immunizations that are based on risk stratification. So let's say, even now, when you talk about genetic high risk, like people say BRCA1 and BRCA2, there are so many more genes that are associated with genetic high risk. And there are so many family histories that are worse than some of the genes, right? So you'll be sitting down and having your risk assessed for so many different things, and everyone is going to have blood drawn, and you will have a risk assessment, and you will be put in a category that will be very defined. You'll have a polygenic risk score done, and it will be determined what category you fall into. And based on what that category is, I would envision you will be given many things to do. You will be given a physical activity prescription, you'll be given this prescription, a dietary prescription, a vaccine prescription, but I cannot see the vaccines I make being given to a 6-year-old. I can see them being given to a 23-year-old or 24-year-old. I would see a vaccine I make extending the fertility time of someone so that they don't have to get an oophorectomy at age 33. They could get it at age 54. I hope someday they could avoid doing that altogether, but I don't see a vaccine that I create going to a 6-year-old.
All right. One thing, Nora, that strikes me is that since you were here the first time, how much work has been done. In your first talk, you really talked about lab research, and now you're really talking about your passion for translational and improving patients' lives. There's so much interest in your vaccines and in your work. Are there trials in which people in this part of the state can enroll in if they're interested, and how might they go about that?
Yes. Our trials are early stage phase 1 and phase 2 clinical trials that are really being run out of Seattle and other academic centers, usually at places like Johns Hopkins or other places. We have good collaborators here in Spokane, like Christine Rinn. Shout out to Christine, who's a fantastic oncologist. And we would like to potentially start some of our trials here in Spokane. So we are working with her to try to, we have some blood draw studies going on, trying to get them open at Christine's site. And if she's interested, maybe try to get some of our trials open there too. So hopefully in the future, we could have trials if Christine will keep working and not retire.
All right, I just got a question from online, and that has to do with how highly genetic is bladder cancer and how much work and study has been done with vaccines regarding bladder cancer.
Our bladder cancer vaccine is pretty far along. And bladder cancer is another highly mutated cancer. It responds well to immune therapy. Our bladder cancer vaccine is directed to early stage bladder cancer. And the reason why we developed it is because the treatment for early stage bladder cancer is a drug called BCG, and it's an instillation drug that is very difficult to be produced right now, and the group that produces it is not really producing it anymore, and it's going to be a worldwide shortage of BCG. And early stage bladder cancer is a commonly recurrent cancer. So we hope to have our bladder cancer vaccine into clinical trials within probably the next 2 years. So it's a top priority for us because we know it's an unmet clinical need.
That's great. I just have one other comment from online, and I'm just going to read it directly. Can you please have her back again?
Oh, that's so nice.
This person had been at your prior talk and, again, as I mentioned, was just impressed by the leaps and bounds with which you're moving and just really appreciate that work. Before we wrap up, I want to once more thank our sponsors, the Spokane County Regional Health District, Spokane County Medical Society, and the University of Washington Public Health Initiative. I do want to let you know also that this is an ongoing series, and our next installation of this series is going to be on January 28th. And we're going to be talking about diabetes mellitus and continuous glucose monitoring. So keep your eyes peeled for that. And once again, just want to thank Dr. Disis for coming out, spending time with us, and sharing this exciting area of research that's going to result in great things. And I think she's going to get that prize. So thank you.